Hello and good afternoon to Washington. First, I would like to thank the organizers for the opportunity to present here. Unfortunately, I cannot be with you in person this year. However, I am very confident this will change in the near future. I'm grateful that the organizers have combined this in-person event with the possibility to attend virtually. It gives me the opportunity to participate and be a part of this great community even when traveling restrictions keep me in Europe. My name is Michael and I work as Chief Technology Officer at SWA Life Science in Sweden. It's my pleasure to present our brand new iLite reporter cell platform for the quantification of anti-AAV neutralizing antibodies. It is the first time we show this dataset. Following our name, SWAR, which means answers in Swedish, we are providing answers to facilitate biopharmaceutical development worldwide. The area of bioassays is a very central part of our activities, and today I will provide some insights into our latest developments. During the last years, we have been extending our activities in the area of gene therapy. Tailored reporter cell lines can be used for a variety of gene therapy applications like potency assays or immunogenicity assessment. Today, I would like to focus on immunogenicity assessment in the context of gene therapy. I will start with a very basic introduction into the area of gene therapy. It is sometimes referred to as the medical breakthrough of the 21st century, since there are some stunning medical results. Today we have approved clinical applications utilizing both the principles of ex vivo and in vivo gene therapy. Very often, adeno-associated viral vectors are used, and it is accompanied with several challenges and hurdles. One of these obstacles lies in the pre-existence or development of anti-AAV antibodies. Focusing on the need for a reliable bioassay for the detection of neutralizing antibodies, we have taken our iLite reporter cell technology and developed a new modular platform for serotype-specific detection of AAV neutralizing antibodies in humans. I will show first results how this platform did pick up AAV2 neutralizing antibodies in individual serum samples. In the end, I will sum up my talk and leave you with a short take-home message. I hope you are as excited as I am, so let's dive into gene therapy. As mentioned, gene therapy is often deemed to be a hope for our future, the medical breakthrough of the 21st century, and there were some groundbreaking medical achievements. But let's start with the question, what is gene therapy? It refers to a genetic modification of cells to produce a therapeutic effect or the treatment of a disease by repairing or reconstructing defective genetic material. The term is also used in the context of introducing a gene into a cell for adding new features, like, for example, introducing a chimeric antigen receptor into a T cell, generating a so-called CAR T cell with newly acquired very specific anti-tumor activity. The history of gene therapy is going on for some decades. However, the first successful nuclear gene transfer in human is a bit over 30 years ago, and the number of clinical trials based on gene therapy is constantly growing. In 2017, 2 gene therapies were approved by the FDA. Spark Therapeutics Luxturna, which treats a mutation-induced blindness, and Novartis Kymriah, which is a CD19-directed CAR T-cell approach for the treatment in the area of hematologic malignancies. In 2019, Avexis Zolgensma was approved for the treatment of spinal muscular atrophy. And I think it is a fair statement to say such drugs generated a wave of high interest and hope due to their medical results. Think about it, blind children reverting partially their sight and newborns with a very rare and commonly fatal mutation for defective muscular development suddenly survived this disastrous disease. Looking at gene therapy today, one can distinguish two forms: ex vivo gene therapies and in vivo gene therapies. In ex vivo gene therapy, you take patients' own cells, known as autologous, or healthy donor cells, known as allogenic, and those are modified outside the body. A viral vector is used to integrate and express a particular protein, such as a chimeric antigen receptor. Kymria and Yescarta are two examples for such ex vivo gene therapies. In so-called in vivo gene therapy, a vector, which is typically a virus, is introduced directly into a patient. The desired biological effect is created by passing the genetic material, for example for a missing protein, into the patient's cells. Here it is often adeno-associated viral vectors. Here are two examples. Luxturna is an AAV2 containing the cDNA for RPE65, and Zolgensma is an AAV9 containing the transgene for SMN1. Besides lentivirus, adeno-associated viral vectors are often used for gene therapy approaches today. AAV are not pathogenic and perform well in transducing a variety of cells. They come in different serotypes and exhibit different tropisms. They can enable long-term expression of the gene of interest, and high-titer vector preparations can be obtained. The cloning capacity is limited to 4.7 kilobases. However, there are some challenges and hurdles when it comes to AAV-mediated gene therapy. It might become very complicated with multigenic disorders like diabetes or heart diseases. In addition, viral vectors, including all the components of a production, may produce severe toxicity. Gene therapies are capable of breaching the Weismann barrier, which would modify the germline, and of course this is something to be avoided. And in general, insertional mutagenesis, for example into a tumor suppressant gene, may induce a tumor. Another challenge lies in the pre-existence or de novo induction of an AAV-mediated immune response, which would reduce therapeutic effectiveness. At the same time, the often short-lived nature of the therapeutic effect might require several treatments. And last but not least, from an economical perspective, gene therapies are among the most expensive treatments in the world. Luxturna, the price of the treatment is close to $500,000 per eye, and several treatments are needed. And for Zolgensma, it's just one shot, but this one treatment is priced at over $2 million, and it is the most expensive drug in the world. Looking at the last 3 bullet points, an assessment for a pre-existing or newly acquired immunity against the AAV vector seems necessary. Regarding the immune response to AAV, up to 90% of the human population has been exposed to wild-type AAV, very often in early childhood, which can lead to pre-existing immunity. It is known that a pre-existing immunity can prevent or result in a loss of transgene expression following AAV administration. Neutralizing antibodies during administration of recombinant AAV vectors limit long-term transgene expression in human subjects. Taking together, an assessment of pre-existing or de novo raised neutralizing AAV antibodies is beneficial and required before during and after treatment with recombinant AAV vectors. In the next part of this talk, I would now like to introduce our cell-based reporter gene technology called iLite. iLite cell-based assays focus on the biological functionality rather than the binding interaction, and this means they can, for example, mirror the mode of action of a particular drug. We commonly pick a certain signaling pathway and we hijack this pathway to engineer signaling into a specific expression of a firefly luciferase. By using chimeric transcription factors combined with synthetic promoters, we achieve a very high pathway specificity. A second luciferase under the control of a promoter for a household gene is serving for normalization. The iLite cells are provided in a very convenient thaw and use assay-ready format. This format is significantly decreasing timelines while offering more reliable and robust results. In short, as depicted here, you take the frozen cells, thaw the cells, pipette the experiment, incubate for several hours, and you have your outcome most of the times on the same day. Our iLite cell-based assays are broadly established, supporting the entire drug discovery and drug development chain. Pathway activation and screening of drug candidates can be done during drug discovery. During drug development, iLite cells are often used in potency assays and for batch release testing for biopharmaceuticals and gene therapies. They are also established for the determination of neutralizing antibodies and for therapeutic drug monitoring. In the field of biosimilar development, iLite can serve as platform for the required comparability experiments. We have now taken this technology and we have developed a novel two-cell line platform for serotype-specific detection of neutralizing antibodies against AAV. Here is a general overview on how the system works. We have developed the system for AAV2 first, however, the modular system also allows for additional serotypes. It is a two-cell line setup with packaging cells and AAV-responsive reporter cells. The AAV packaging cells contain a genetic element which is packed into a serotype-specific capsid and acts as a transcription factor. It binds to a synthetic promoter for specific activation of luciferase expression in the AAV reporter cells. On the left-hand side, you can see the process for the titration of packaging and reporter cells. I will show this step in more detail on my next slide. Once the optimal ratio of packaging and reporter cells has been identified, the neutralizing assay can be performed. In the neutralizing assay, packaging cells and AAV-responsive reporter cells are co-cultivated. Neutralizing antibody standards in serum samples are added and incubated overnight. The resulting luciferase expression is proportional to the level of AAV transduction. The inhibition of AAV transduction can be calculated, and the presence of neutralizing antibodies against AAV can be determined. So, let's take this step by step. First, I will zoom in on the activation assay. Here, the ultimate goal is to determine the optimal ratio of packaging and reporter cells for the new batch of packaging cells. This is something which needs to happen one time per batch of packaging cells. We take a constant number of reporter cells and cultivate them with a variable number of packaging cells overnight. The firefly luciferase expression is proportional to AAV transduction. On the right-hand side, you can see the sigmoidal dose-response curve, the number of packaging cells against the relative light units measured as a luciferase activity. The optimal ratio of packaging and reporter cells is calculated. You can see it in red. We currently use the EC70. It is a good compromise to be within a certain linear range, and at the same time it offers a good dynamic range of the overall assay. This ratio can now be used for the upcoming neutralizing assay. So now let's have a closer look on the neutralizing assay. The packaging cells and the reporter cells are co-cultivated in this optimal ratio identified before. And now we add controls, we add a neutralizing antibody standard, and we add individual serum samples and incubate overnight. The next day, the luciferase expression is proportional to AAV transduction. The neutralizing standard is a pooled human serum, which is depleted from human IgG. This depleted serum is used as matrix. Enriched and characterized IgG, it is the approved drug known as QVIC, is spiked into this matrix in increasing concentration. Since QVIC is a pool of IgGs from many different people, it naturally contains AAV neutralizing antibodies. In the graph on the right-hand side, you can see the concentration of QVIC in microgram per milliliter on the x-axis against the relative light units on the y-axis, and the percent inhibition is indicated as well. You start with a high signal since no AAV neutralization is detected with low concentration of antibodies. With increasing concentration, the signal is inhibited down to full inhibition with a certain amount of the IgG pool. Now we can calculate inhibition of AAV transduction and analyze serum samples for the presence of AAV neutralizing antibodies. Before showing the individual samples, we also tested for serum tolerance. We did the experiment explained on the last slide and used various amounts of serum or matrix, as I said before. Serum tolerance was acceptable up to approximately 25%, so we decided to work with 10% human serum. Serum dilutions of 1 to 10 are very common. So keep in mind all experiments in the upcoming slides are done with dilutions of 1 to 10, 10% human serum. Here you can now see the results after testing 20 individual human serum samples. This was healthy population. All the samples were measured in triplicates. The colored lines represent the average of these triplicates. 0 to 100% inhibition was added from the standard curve. As you can nicely see, a broad range of anti-AAV2 inhibition could be observed. In the next experiment, we now picked 4 representative samples. The green one representing maximum inhibition, the blue one with a very high inhibition, the violet one with a very low inhibition, and the pink one, which is on the top, with minimum inhibition. In order to analyze the inhibitory effect observed, we diluted these 4 individual samples. Starting point was at 1 to 10, which is the same dilution as used in the original experiment with all 20 samples. You might remember I mentioned before we tested for serum tolerance, and as a result we decided to continue working with a 1 to 10 dilution, meaning 10% of human serum. In this experiment, in addition, we added dilutions of 1 to 50, 1 to 500, 1 to 2500, and 1 to 12,500. In the graph on the right-hand side, you can see the results. The green sample was the sample with the maximum inhibition. It stays maximum with a 1 to 50 dilution, and the inhibition goes down at 1 to 2500, but it's still visible. In blue, the inhibition stays up until 1 to 500. In violet, which was the low inhibition, it falls to 0 inhibition already at 1 to 50 dilutions. And in pink, the minimum inhibition stays at no inhibition, as one would expect. So this means the inhibitor capacity could be reduced by serial dilution of the serum samples, and the samples exhibiting higher levels of inhibition needed a higher dilution to reduce this inhibition potential. So much for the data set I wanted to show you today. Our lab is currently working on generating data to confirm robustness of this assay by repeating the assay with different cell batches on different days with different operators. And I would like now to sum up this presentation and provide you with a short take-home message. The data we have observed in our laboratory so far suggests that our new iLite AAV neutralizing platform can be used to screen sera for neutralizing antibodies to recombinant AAV vectors. It allows for a serotype-specific detection of neutralizing antibodies. AAV2 is available. AAV5, 8, and 9 will be available soon, so we are planning to complete the portfolio of standard serotypes in the future. In addition, its modular system allows for custom solutions adapting to sophisticated hybrid or chimeric capsids or synthetic capsids. And some words on the assay. The assay setup is very easy and very quick. It eliminates the need to purify the vector between the 2 steps because the cells are co-cultivated and the cells are available in a frozen, thaw and ready to use format. As a take-home message, I would suggest following bullet points. I think we all agree gene therapy offers promising possibilities for the development of new treatment options. However, it comes with some hurdles and challenges, and pre-existing as well as de novo generated neutralizing antibodies against the viral vector can be problematic. They can impact the therapeutic potential, and there needs to be an assessment of such neutralizing antibodies. The iLite reporter cell platform presented in this presentation offers assessment of anti-AAV neutralizing antibodies. It is a novel tool for rapid and reliable detection, and we developed it to be used in clinical applications. The proof of concept data shown was for the detection of anti-AAV2. Further serotypes are currently under development. However, please keep in mind the modular system of this platform can also be utilized to design tailored solutions for neutralizing antibody assessment. Viral vectors with hybrid, chimeric, or semisynthetic capsids could also be measured using this technology. In the end, I would like to thank our exceptional scientists sitting in Malmö and in Paris for developing this great assay and for generating this nice set of data. And of course, I would like to thank you for staying with me during this presentation and for your attention. Please feel free to reach out for questions and comments at our booth, on our webpage, or via email. SVAR is very much looking forward to also providing answers to your challenges in life science.